I'm a corneal specialist at UT Southwestern Medical Center in Dallas, and I treat disorders of the cornea. The cornea is the clear, transparent tissue in the front of the eye. And just a basic primer, the surface of the cornea is the epithelium. It turns over fairly rapidly. It's about 5 to 7 cell layers in depth. It sits on a collagenous stroma that makes up the bulk of the clear tissue. And this stroma is sparsely populated by keratocytes that are thought to turn over in the months to years range and deposit and maintain the clear collagen structure of a very regular periodicity that gives it its transparent structure, a prerequisite for vision. And the layer of interest is the inner layer of the cornea, the corneal endothelium. And those are the pump cells, as I explained to my patients. And there's a normal attrition of these pump cells. They're of neural crest origin, and there are probably about 6,000 cells per square millimeter at birth and in infancy. And there's a slow attrition. In middle age, the cell density goes down to about 2,000. And if it reaches less than 500 cells per square millimeter, the cornea has become edematous. And that's when patients become symptomatic. So I was recruited to UT Southwestern about 20 years ago to implement new types of corneal transplant procedures for patients with this common age-related corneal disease called Fuchs dystrophy. It affects about 4% of whites over the age of 40. And for unknown reasons, there's a strong female bias. And with no medical treatments, patients that develop significant disease go on to needing corneal transplantation. And we've come a long ways in terms of offering corneal transplants to our patients. 20 years ago, we had to replace the full thickness of the cornea, but now we're able to just selectively remove that inner layer of the cornea and transplant just the inner layer of the cornea. So we really have improved our patient outcomes with these newer modalities of corneal transplantation. So this is a clinical spectrum. Any of you that have gone to the ophthalmologist know that binocular slit lamp, the microscope that's used to examine the front and back of your eyes. And when we examine patients with Fuchs dystrophy, we're able to see these focal excrescences on retroillumination, those little specks on the upper left-hand corner. It's just a reflected light. And these little specks are the thickened basement membrane focal excrescences that are deposited by the endothelium. And early in the disease course, patients notice glare. And with moderate disease drop off of those endothelial cells, the cornea has become edematous. We rarely let patients get to the latter stages of the cornea. Bottom right is when it's completely neglected. We never really let patients go on to that stage. We intervene surgically. I really think of Fuchs dystrophy as a disease of accelerated senescence. This is a post-mitotic layer of neural crest origin. There's normal attrition and normal thickening of the basement membrane that goes on with age. But this thickening of the basement membrane, which is central to disease pathogenesis, is really accelerated grotesquely in patients with Fuchs dystrophy. And you get those focal excrescences, gutai, that are pathognomonic on the slit lamp examination. So there's no doubt that there's an upregulation of extracellular matrix genes. Unfolded protein response is likely playing a role. And like many neurodegenerative diseases, mitochondrial dysfunction has been implicated. Epithelial mesenchymal transition is likely playing a role. And ultimately, you get loss, programmed cell loss, apoptosis. And when you lose enough of these cell layers, pump cells, you get an edematous cornea. The genetics of Fuchs dystrophy has really advanced a lot over the last 20 years. My clinical colleagues, John Gotch, 20 years ago, studied his large families from Hopkins, and he identified a chromosome 18 as being important using traditional linkage studies on large families. In 2010, my former colleague, Al Edwards, who was at UT Southwestern, he moved on to the Mayo Clinic, and he did a very important genome-wide association study that highlighted a 5' single nucleotide polymorphism in this TCF4 gene, or the transcription factor 4 gene. And this is an ubiquitous transcription factor. It's highly expressed in CNS tissues. At the time, we knew it based on human genetic studies, Pitt-Hopkins syndrome. When you lose one copy of this gene, you get this very devastating neurodegenerative phenotype with motor and intellectual disability, seizures, hyperventilations. but there were no associated corneal phenotype. Myopia and cataracts had been described. So I think even at that point, we knew, I think based on human genetics, that haploinsufficiency was unlikely playing a role in disease pathogenesis of Fuchs dystrophy, this age-related corneal disease that I have an interest in. And then a neuropsychiatric group at Hopkins in 97 used this repeat expansion detection assay, and they found this locus on the TCF4 gene on an intron that was unstable in their Caucasian pedigrees when they looked at DNA samples from the CEU cohort of DNA samples. And they tried to see if this intronic trinucleotide repeat was associated with bipolar disease or schizophrenia. It wasn't. So they identified the repeat locus, but didn't know what the phenotype was. Subsequent studies at the Mayo Clinic in a small cohort saw that trinucleotide repeats in the intron, CTG repeats in the intron 2 were strongly associated with Fuchs dystrophy. But about 3% of controls also had this intronic trinucleotide repeat without disease. So it wasn't quite clear, was this intronic trinucleotide repeat just a microsatellite in linkage disequilibrium with another nearby causal mutation, or was it a causal mutation with incomplete penetrance? So 2012, I launched my human genetics lab at UT Southwestern. I was telling one of the organizers, I think I had an early midlife crisis and I wanted to do something more than just see patients and operate. And inspired by my brother, I wanted to do something in the human genetics field. And at that point, I had a lot of patients and families. And we quickly replicated the strong trinucleotide repeat association with the Fuchs phenotype. But we also saw that there were cases of incomplete penetrance. We looked at it in our families. And we also did trans-ethnic replication studies. In whites, this trinucleotide repeat confers a 30-fold increased risk of disease. In Singapore Chinese, it confers a 60-fold increased risk for disease. So it was really with these trans-ethnic studies that we became pretty confident that this was likely a causal mutation. And about 70% of whites in the US have this trinucleotide repeat in the TCF4 gene that have Fuchs dystrophy. The first question we wanted to answer is there haploinsufficiency of the TCF4 gene? At least at the mRNA level, looking at a constitutive exon 18, which has the basic loop helix domain that's important for transcription factors, we didn't see any evidence of a decline in the mRNA levels. I think there's likely a more complicated story as to the isoforms. There may be a shift away from some of the isoforms that express the 5' exons based on subsequent works that have been published. And really inspired by work in the myotonic dystrophy field, we really wanted to test the hypothesis that repeat RNA, CUG repeat RNA, was accumulating in the endothelial cells of patients that were undergoing corneal transplantation. And probably the high point of my scientific career to date was, looking at these patient corneal samples and using the FISH assay that had been previously described for myotonic dystrophy and seeing these red dots, these foci lighting up the corneal endothelial samples. And, I've been very much inspired by the work done in the myotonic dystrophy and the work done by many in the audience. And Joel Gottsfeld's group also detected these repeat RNA foci, and he elegantly showed co-localization of the splicing factors MBNL1. Two have also been shown to be co-localizing, and our working disease model is one of MBNL sequestration resulting in mis-splicing of target exons of MBNL. These foci are certainly specific for CTG repeats. If you look at a patient with just corneal decompensation from glaucoma or another cause, you don't see foci. If you look at patients with Fuchs dystrophy that don't have the trinucleotide repeat expansion in the TCF4 gene, you don't see foci. So it's specific for the repeat expansion. And so all the samples up until this slide that had foci had the trinucleotide repeat expansion in the TCF4 gene. I work closely with the eye bank at UT Southwestern, and patients whose corneas have Fuchs are not eligible for transplantation. So, we like to use those donor corneas from the eye bank for our research. And we ran across a pair of donor corneas from the eye bank that had foci, and we genotyped the trinucleotide repeat locus in the TCF4 gene, expecting to see a trinucleotide repeat expansion, but we did not. And this was really vexing me and perplexing me. And I was in India at the time, and I called or emailed my tech and said, you know what, we need to genotype this donor for the DMPK repeat. And we had tissue from this donor, and we did triplet repeat prime PCR and detected a repeat expansion in the DMPK locus in this donor cornea with Fuchs that didn't have the trinucleotide repeat in TCF4. So this was really cool, we thought. And we working through our iBank, we reached out to the next of kin of this donor. They had a clinical diagnosis of myotonic dystrophy, DM1. They had southern blot analysis that confirmed the DMPK repeat expansion. And so here, we demonstrated really a co-convergence of two different trinucleotide repeats that result in foci formation and degeneration of the corneal endothelium. In a fairly small cohort of patients with myotonic dystrophy, about nearly half of them, if you look very closely, had some findings of Fuchs endothelial corneal dystrophy. It's unlikely for the DMPK locus to be contributing a huge amount to our overall the genetic burden of Fuchs. We looked at about 300 patients in my cohort. 70% had the repeat expansion in TCF4. But interestingly, we did find one patient that had the DMPK expansion and not have a clinical diagnosis of DM1. Perhaps their repeat expansion was not significant enough to cause the muscular, cardiac, and CNS phenotype. But so here, I think it's a cool story because it's one repeat CTG and a non-coding gene. There are two different genes, DMPK as well as TCF4. And what they have in common is the repeat RNA that accumulates in the affected tissue, and both can cause corneal endothelial disease. But we've done some quantitative studies, and most corneal endothelial cells have about 2 or 3 of these foci. And there's probably a one-to-one correspondence between the intronic RNA molecules of the intron of TCF4. So we feel that there are a relatively few number of RNA repeat molecules at any one given time that are likely sequestering both MBNL 1 and 2. We've done some quantitative studies on MBNL, both RNA and protein. I think in this corneal endothelial tissue, the copy number of MBNL is likely much less than in proliferating cells. And if you further look at their nuclear to cytoplasmic distribution, I think a lot of the MBNL is likely in the cytoplasm. So only having a few repeat RNA molecules in the nucleus, it's plausible that they're being functionally sequestered. And work done at the Mayo Clinic suggests, at least in cultured cells, that there are somatic mutations that are going on in the corneal endothelium, that the repeat number may be much larger in the tissue of interest compared to peripheral leukocytes. So this is our working model of Fuchs dystrophy, a intronic CTG repeat expansion, the TCF4 gene. We've looked at donor corneas that have the repeat locus without findings of Fuchs dystrophy. 4% of whites have the repeat. So we're able to see effects of foci and mis-splicing, even in asymptomatic donor corneal tissue. So we think that those are important in early disease pathogenesis. If you look at the transcriptome changes in early and late stage disease, there's an upregulation of extracellular matrix genes that precedes the activation of fibrosis, mitochondrial dysfunction, and immune cells that are ultimately at play at end stage disease. In collaboration with the David Corey lab, we've tried to develop at least proof of concept experiments and projects demonstrating that antisense oligonucleotides, duplex RNAs, and even CUG repeat targeting gene therapy with an inactivated Cas9 can block the foci formation and mis-splicing. So for the remainder of the few minutes that I've got on the podium, I'd like to share with you this recent body work that's been published. We're very keen on what other cells may have foci in the eye. What drives this cellular specificity for the foci formation and disease formation? And, as an active cataract surgeon and corneal transplant surgeon, I have access to the corneal endothelial tissue as well as the lens capsule. The lens, when it gets cloudy, is a cataract. And that's one of the most commonly performed surgeries in the U.S. And all of you know that almost virtually every myotonic dystrophy type 1 patient will go on to develop cataracts. We don't quite see that, though, in Fuchs dystrophy. We certainly don't see the iridescent Christmas tree cataracts in Fuchs dystrophy caused by the TCF4 repeat expansion. But I have access to these tissue samples, and through the eye bank, we're able to look at all the layers of the cornea in donors that have the trinucleotide repeat that we genotype. And additionally, in the donor corneas, we have access to the trabecular meshwork. This is part of the eye is important for the aqueous outflow. It's diseased in patients with glaucoma. So we were also keen to see if there's foci formation in these layers. Interestingly enough, when you look at the lens capsule of patients with the TCF4 repeat expansion, you don't see foci. So that kind of coincides with the absence of the polychromatic iridescent cataracts that are seen in myotonic dystrophy patients. We don't see the foci. When we look at the other corneal layers, the far right, that label 5 is the endothelium. That's what's stripped and replaced in endothelial keratoplasty. And about 80% of surgical samples have foci, and 80% of the donor corneas from the eye bank also have foci in this layer of interest. But the donor corneas also give us access to these other cellular layers. If you look at the epithelium, that's the surface cells on the cornea. They turn over very rapidly. Within 5 to 7 days, they're turned over. We don't really see foci in that cell layer. In the substance of the cornea, the collagenous stroma, or the keratocytes that populate this clear transparent tissue, and they form the extracellular matrix. There's very slow turnover. Interestingly, they're also of neural crest origin, similar to the corneal endothelium. And we see about 10% of these cells have foci, compared to about 90% or 80% of the corneal endothelial cells. So it affects all the layers of the cornea. We've done in vivo confocal microscopy that shows that all the layers of the cornea are affected in patients with the trinucleotide repeat and TCF4 if you look hard enough. And then finally, we were quite excited to see that cells of the trabecular meshwork, this key tissue that is important in glaucoma, it's also of neural crest origin, and it may have a similar stem cell to the corneal endothelium. And about 40% of these cells also have foci. If you look at target exons of MBNL, like INF2, MBNL1 and 2, the corneal endothelium has the most robust evidence of mis-splicing of these target exons. You really don't see the mis-splicing in corneal epithelium or the stroma, but you do see some evidence on the very bottom, or the mis-splicing signature of some of these MBNL sensitive exons in the trabecular meshwork. So the mis-splicing seems to correlate to the burden of foci, which may not surprise this audience much, but was interesting to document. When you look at overall gene expression, we chose some genes that we knew were going to be upregulated both early and late in the disease course, differential gene expression of some extracellular matrix genes. And the upregulation of these extracellular matrix genes and others that we had previously documented in Fuchs in the corneal endothelium were primarily seen in just that layer. We really didn't see upregulation of gene expression of these other fibrosis genes in the other cell layers of the eye that we examined. And then trying to figure out what is driving foci formation and mis-splicing in certain corneal layers and tissues of the anterior segment, we went back to looking at the overall expression of TCF4. So we've used exon 18 as a surrogate since it's a constitutive exon, and there are over 40 transcripts of TCF4, and many of which don't include the 5' end that includes the intronic repeat. But when we look at a constitutive exon of exon 18 that's found in all TCF4 transcripts, you see very high levels in both corneal endothelium and trabecular meshwork cells, and some modest expression in the stromal keratocytes. Those are the cells that we see foci. But if you dive a little deeper and you look at exons that are on the 5' end that actually harbor the repeat, you really see much stronger expression in corneal endothelium and trabecular meshwork, and less so in the other cells. So we feel that, for the foci to be present and cause their toxicity in the anterior segment of the eye, you need to have not only expression of TCF4, but those transcripts that harbor the 5' end with the repeat expansion. So in summary, we feel that the CUG repeat RNA that we've characterized both in patients with a trinucleotide repeat in the TCF4 and DMPK, they predispose them to CUG repeat accumulation in their corneal endothelium, and these foci, likely through an MBNL sequestration model, predispose them to corneal endothelial disease. About 80% of corneal endothelial cells have foci compared to about 40% of trabecular meshwork, about 10% of stromal cells. Embryonic lineage may be playing a role in terms of how which cells express TCF4 and which isoforms are expressed, because corneal endothelium, meshwork, as well as stromal keratocytes are all of neurocrest origin. With a few exceptions, most of the mis-splicing is seen in the corneal endothelium. And we feel that expression of the repeat sequence, especially the 5' end of the TCF4 gene locus that has the intronic repeat, likely drives the formation of the repeat RNA to be able to exert their pathological consequences. And likely these cells may be more prone to somatic mutation based on the work of others. I'd like to acknowledge past students in my laboratory, as well as my collaborator, David Corey. Chao Jing is at the McDermott Center. I've collaborated on all of our human genetics work with my colleague at the McDermott Center. I'm grateful for the funding that I have, and want to make a plug for my lab. I have a very small group, but whoever might want to join us will be given right projects that are very close to being brought to the finish line. I welcome any interest of potential postdoc candidates.
Thank you for the talk, Dr. Mootha. From your research, it's very interesting to see the trinucleotide repeat in TCF4. It's uniquely expressed in corneal endothelium and also in the trabecular meshwork, which might explain that some FECD patients also have glaucoma. And so we know the FECD is a genetic heterogeneous disease, which is also related to multiple environmental factors. So do you think the trinucleotide repeats in TCF4 works with other gene mutations and all work with environmental factors? And how do you think the TCF4 plays in the FECD disease?
Yeah.
Thank you.
Yeah. I think TCF4, it's responsible for 70% of the genetic burden of Fuchs. There's no doubt that other genes are involved, including DMPK in the other 30% or subset. So there are other genes to be identified. In my opinion, the repeat locus in itself is sufficient in terms of a genetic insult, but you bring up the important point of environment. And I think there's good epidemiological data that smoking, perhaps UV light, may also be environmental risk factors. So I think there is likely a gene environment interaction. But in my opinion, I think the trinucleotide repeat in the context of either TCF4 or DMPK may be sufficient in terms of the genetic insults required.
So
we are a little tight on time. I was just wondering, you showed that the TCF4 foci weren't present in other regions of the eye, in particular, or at least in the lens, so that maybe explains the potential lack of cataracts. But have you looked in other tissues? And again, the obvious question is why aren't these guys getting other
DM-like I'd like to think I'm an astute clinician. I think the disease is primarily one of the eye. I don't see, CNS or skeletal cardiac issues in my patients. That they live into their 80s and 90s. We've not had access to other tissue. I have access to my corneal samples and the eye bank tissues, but it would be very interesting to kind of map out globally, in a donor whole body, looking at, the burden of foci formation.
Hi, I was just wondering, have you looked at comparing, like when you remove the corneals and look at the actual repeat burden that's in that tissue? Is it different than like what you see in the patient's blood? Have you checked that to see if it was
we're trying to do that, but the amount of DNA is very limited. We've had a lot of issues with some of these more sophisticated small pool PCR or rolling circular amplification or these types of assays. For PacBio, you need a little bit more DNA than what we can achieve with our corneal sample. So right now, I think there are some limitations in terms of actually looking at the repeat number in tissue. We have some ideas, but it's likely probably much greater in that layer than in peripheral leukocytes. And at least in cultured cells, others have shown that the repeat in cultured cells from that patient's corneal endothelial samples is much higher than peripheral leukocytes. But we'd like to very much look at it in the tissue of interest. It's likely much larger than in peripheral leukocytes. Yeah.
I think we're in good shape.